So first of all, we're going to talk about cellular respiration, and we're going to be talking about aerobic respiration where oxygen is needed. We'll also then briefly talk about anaerobic respiration and then fermentation. So you need to be able to compare these three processes and kind of understand why they occur in the cells that they do. And so when we look at the macromolecules, our lipids, carbohydrates, proteins, nucleic acids, the carbohydrate glucose, like I mentioned before, is most easily broken down. It's easiest to obtain energy from that. And so most of the models that we look at glucose. You could get energy from other molecules too, but we use that as our primary example. In cellular respiration, we're breaking it down into three stages: glycolysis, the citric acid cycle, and the electron transport chain. Some textbooks you see will break it into 4, but it still has the same processes in there. So we'll go with your textbook and use 3. And so what is the goal of cellular respiration? Well, it's to break down these macromolecules. So if you think of us, when we eat food because we need to obtain energy from that food. How do we obtain energy? We break things down to the simplest molecules like glucose, and then that can be used in order for all of our cells to actually generate the ATP. And so obtaining energy is the goal. This is not exactly a simple process, as you can see here, but if you can kind of follow along with the basics, I think that's a little bit helpful. And so if you look here, the first reaction is called glycolysis. And you can look at the diagram to the right. It's a little confusing, probably, but I'll try to simplify it. And so first of all, if we begin with glucose, when we look at glucose, glucose has 6 carbons, and you can see that in the formula here, C6H12O6. We're basically just going to see a rearrangement of these molecules, and we're going to see a production of energy at the end. And so glycolysis is just we're starting with glucose, we're breaking down glucose a little bit, not all the way, and we're ending with 2 pyruvates. And so the pyruvate molecule is shown down here. What's really most important for you to understand is that there are 3 carbons in there, and there's actually 2 of them, and so we would have 2 pyruvate molecules. So as far as our carbons, if we're kind of keeping track, we can still figure out where those original 6 carbons are. In a cell, regardless of whether it's a prokaryote or a eukaryote, this process takes place in the cytoplasm. It does not require oxygen. If oxygen's around, it's not going to use it. If it's not around, it's not going to use it, of course, as well. And then the goal here of this stage is to generate some ATP. And so it actually generates 4 ATP molecules for 1 molecule of glucose, but it required to put in 2 at the beginning, so we actually have a net gain of 2. And when you look at these figures, if you can just see, oh, here's some ATP that was put in, there's 2, and we got 4 ATPs out of it. So you can kind of find what I'm talking about in this diagram. Also produced here are 2 NADH molecules, and these act as electron carriers. That's really about all you need to think about now. And so there's an NADH, this is just a compound, and it actually acquires protons and electrons. So when we see it as NADH, we know that it acquired these electrons and protons. And you can think of these as kind of bundles of energy. The NADH is going to carry it temporarily, and then later it's going to unleash those electrons in order to generate ATP. So that's the first stage, glycolysis. The second stage is called the citric acid cycle, sometimes called the Krebs cycle. And so we have those 2 pyruvate molecules that we started with, and they're going to be broken down into 2 acetyl-CoAs. Now those and 2 of those only have 2 carbons each, so we don't really see our 6 carbons anymore. But what happened to the other 2 carbons is they actually combined with oxygen and produced 2 carbon dioxide molecules, and these are released. And we'll see carbon dioxide in our overall equation at the end, and hopefully that'll help it make a little more sense. But you may have to go back over this a couple times. There's also a 4 carbon molecule that temporarily gets used, and so it gets introduced. It combines with those 2 carbon molecules, so we get back to 6, which seems a little frustrating, but it's really just a temporary molecule in order to let things go through the citric acid cycle. And so we'll kind of disregard that. It'll break off later in the cycle that you'll see. And so what we end with after this stage is basically, this happens twice. It goes through 2 cycles of the citric acid cycle, and all the carbons are now broken down. So that original molecule is totally broken down. we get 4 carbon dioxides. So those 4 carbons that had been remaining actually combine with oxygen to get carbon dioxide. There's a generation of just 2 ATP molecules. There's a lot more NADHs to carry electrons, and there's also another electron carrier called FAD, and when it combines with the electrons and protons, it's called FADH2. So they carry these high energy electrons, and we're going to see those build up, build up, build up, until finally they're going to be used in the last cycle. So the citric acid cycle looks complicated, and it is. We're not going to worry too much about that. But it takes place, if you're a eukaryotic cell, this takes place in the mitochondria. But if you're a prokaryotic cell, hopefully you remember that you don't have a mitochondria, and so the closest thing is just the cell membrane or the plasma membrane. And like I mentioned before, this cycle actually takes place twice. So anything you would see on here, we would multiply this times 2. And again, it looks kind of complicated, but we have here's our pyruvate to acetyl-CoA. Sometimes this is referred to as stage 2 when we break it into 4 groups, but it doesn't really matter. And so here's the 2 carbons. This enters into the citric acid cycle, temporarily combines with this 4 carbon molecule here, and so then it goes through the cycle. As we go through, you can see we're picking up, here's an NADH, here's an NADH, here's an FADH2, here's another NADH. Again, these are going to carry electrons and be able to transfer them later as these bundles of energy, and the protons are going to be important as well. You can see that we have a carbon combining with oxygen and a carbon combining with oxygen. So that's 2 of our carbon dioxides. Our total was 4. Well, we go through this twice as well. I can see here 1 ATP molecule. When we go through twice, that's our 2 ATPs. And now this oxaloacetate actually just gets recycled back as well. So we really have broken down the carbons from 6 down to 3, down to 2, and then broken this down further so that they combine with oxygen. So I like to trace the carbons. I think it makes it kind of easier to see, and you can kind of go back and look at the diagrams and just look to see where those actual products that I list are. Lastly, we then have the electron transport chain. And so everything that's kind of piled up so far as the electrons on NADH and FADH2 are going to be transferred. And if you remember, they had those protons on there, which is written as an H+. Well, when an H+ is transferred to an O2, then we actually have H2O, which is water. And so that's how we get that part of the equation that you'll see as well. And so this takes place in the mitochondria if you're a eukaryote, or the cell membrane if you're a prokaryote. Lots of enzymes involved. All these little pink figures here are enzymes. We're not going to go into that detail, but just trust me, there's a lot that are involved. And so these electrons just get transferred to these different proteins here, these different enzymes, and eventually they finally get delivered to oxygen, and that's where we get our water. We also have been building up these hydrogen ions, these protons, and these are actually going to help to drive this proton pump. And so this proton pump here is another enzyme. It's called ATP synthase. When this pump gets actually literally moving, because these protons are pumping through it here, then it helps to put ATP together. So remember, enzymes can put molecules together or take them apart. In this case, it's building ATP, and so here's our generation of ATP. And in this case, there's a lot of ATPs that are generated in the electron transport chain. So that's a big thing to remember as well. So it takes a lot to kind of look at this and figure out what's going on, but the more you do, it's a little bit helpful. But I just want you to understand in general what kind of the products are here as well. And so we get a lot of ATP is the point at this point. And so I kind of say 30-something here. Prokaryotes usually get more because they're smaller and they don't have to do that transfer to the mitochondria. They don't have as far to go. And so eukaryotic cells actually lose a little bit of energy and so generate a little bit less. And so a lot of times the number of 38 is used for prokaryotes and 36 for eukaryotes. That can be changed a little bit depending, but that's kind of the number that's most often used. And so here's the overall summary. And sometimes, maybe you've learned this in the past and maybe forgotten about it a little bit too. But basically we have our glucose, and so we have those 6 carbons. In the presence of oxygen, what's going to happen through all those series of reactions that we just looked at, eventually we're going to get 6 carbon dioxide molecules, 6 water molecules, and then ATP, and a whole lot of ATP. That's just looking at one molecule of glucose. And so if you go back and look, you can kind of find where do we see the 6 O2s, or I'm sorry, where do we see the 6 CO2s? Where do we see those 6 waters? Where do we see the energy that happened as a result? And so again, that's called aerobic respiration. That's because oxygen is what finally accepts those electrons and become water. And so there's the water in our equation. If it's anaerobic respiration, then those are organisms that aren't relying on oxygen, but they're relying on something else. And so rather than oxygen being their final electron acceptor, something else is. And it can be a nitrate ion, it can be sulfate, it can be hydrogen sulfide, etc. And so it just kind of depends on what is used and what is your final electron acceptor. But basically it's not using oxygen. So the processes are very similar. The ATP generation's not quite the same, but we can think of it as being close enough for now as well. There's also the process of fermentation, and so we'll look at that a little bit more specifically as well. But basically, if we kind of go back and look at glycolysis, then we don't go much further than that. And so we get a little bit of ATP, the 2 that we saw as well. And so the end products are going to be different. We're not breaking things down completely all the way. And so this is a good summary to look at, figure to look at in your book, just to kind of summarize things. But again, you should just kind of look back and just understand that what are we really doing? We're breaking down glucose. If you follow the carbons, it's just really a rearrangement, and eventually we're generating ATP. And so looking at that overall equation in a little more detail can be helpful. So fermentation. We've already talked maybe a little bit about fermentation, and just that fermentation is a useful process and we get a lot of products from that as well. But as far as what's going on, if we look at something like a yeast cell in this case, remember that's a eukaryote, and Saccharomyces cerevisiae was an example of that that's used quite a bit. Basically, if we look at glucose, we go through glycolysis. And so basically we get down to pyruvate, but instead of then going through the citric acid cycle, the process is kind of almost over. And so those electron carriers, the NADH, has to actually get recycled. And so pyruvate changes a little bit. In the case of yeast metabolizing glucose, it would be changed to ethyl alcohol. And then also there's carbon dioxide gas that's given off as well. And so we're still starting with that C6H12O6 in this case, and we're just not breaking it down all the way. And so respiration can't take place without oxygen. So if respiration is not possible, then fermentation will take place as well. So not as much energy is generated, but it's still a useful process to be able to use. And again, our cells are able to ferment if we're doing some activity, an anaerobic activity like a sprint or something like that. We can still get a little bit of energy from that, but we can't build that. We can't keep that going forever unless we had oxygen to keep sustaining us through that. And so it kind of depends on what you start with as far as what you can get. So you can start with yeast if you have them metabolize barley. That's actually used to make beer, and metabolizing potatoes is used to get vodka. So really the starting products depends on what your finishing products are as well. And so there's also other organisms. There's even bacteria that does this as well. Human muscle cells also convert pyruvate to lactic acid. And so alcohol is not always the end result, but lactic acid could be as well. And so a lot of times we see this in yogurt, we see this in making cheese. If you think of Swiss cheese, where do those holes come from? Well, it has to do with the gas that's actually an end result as well. So there's a lot of different processes that happen depending upon, again, who you're starting with and kind of what you're ending up with as well, or what you're using to ferment in the first place. And so these are just some examples that are shown there. And again, this kind of reminds you of glycolysis here. We get down to pyruvate, and then we go a little bit further than that, but then we stop. So if we have lactic acid, if we have ethanol, we have some of these other products, they are not completely broken down. And if you think about it, that kind of makes sense because we're just still have some of those molecules in there. So we have some of those chemical bonds in there. So we got a little bit of energy, but it didn't get broken down all the way, which is fine, and if you like and enjoy some of these products that are used here as well. And so looking at some of the products, it just kind of depends, like I said, what you start with as far as what you end up with. And there's a lot of products out there that are, this process of fermentation is used to create. So if you're interested in that, you can just kind of Google products of fermentation. You might be surprised that a lot of the foods that you eat and products that you use are actually produced via fermentation. All right, lastly, we're going to very briefly look at photosynthesis here, only because if you kind of look here at the equation, you'll notice that it's kind of the reverse of respiration. And so we need oxygen. If you're a cell like us, or our cells, we need oxygen. And so where does that oxygen come from? Well, actually it comes from the process of photosynthesis. So it is very important to kind of consider as well. It's kind of a lengthy process also, so I'm summarizing it here in one little slide. But basically, energy from sunlight is captured in pigments, and these pigments are going to then transfer that energy. If you look here, you see electron carriers again. You see them a little earlier than we saw before. But why are they producing ATP? Why is this energy necessary? Well, photosynthesis is also building glucose and building sugars. And so if you look here, to put something together, that can be kind of hard and requires ATP. So you have to first of all generate the ATP. Another thing that's getting generated here is that NADPH. It's a little bit different this time, but same idea. And so that's going to carry the electrons, and that's going to help us to get ATP later as well. And so the carbon dioxide then is used. We have the light at first, and then the carbon dioxide and the light combines with water. I kind of didn't tell you that up here, but that's where the water comes into the equation here. That's how we get the oxygen. And so then carbon dioxide gas with ATP and NADPH is used to put these carbohydrates together. And so it's basically called these carbon trapping reactions here. It's called different things in different textbooks as well. But basically we're putting together this molecule, and that molecule is glucose. So any organism that's capable of photosynthesis can make glucose, can actually use that glucose for their own metabolism, because these organisms have to be able to ferment or respire. And they can also be stored as starch for future use, or then, if it's something like a plant comes along and we eat it, and we are breaking down that glucose and using that glucose for later.